Hello and welcome. I am Miss Stephanie, and in this video we are going to learn about Punnett squares. Punnett squares are used in genetics to determine the possible combinations of genes that will occur at fertilization. It consists of squares, which are used to create a combination of genes and predict the possible genotypes of the offspring. In this video, we will only learn about monohybrid crosses, and in a subsequent video, we will talk about dihybrid crosses. A monohybrid cross is a cross between a single paternal and a single maternal gene. For our example, we are going to use height as our gene. Tall will be the dominant trait, represented by a big T, and short will be the recessive trait, represented by a little t. Before we can start talking about how to do Punnett squares, we need to understand a few terms first. The first term we need to know is genotype. A genotype is the letter representation of a person's gene. The genotype is consisted of two letters, either lowercase, uppercase, or a combo of the two. The next term is phenotype. The phenotype is the physical representation of a genotype, so being tall, or the color of your hair. The next word is dominant trait. A dominant trait is one that only needs one allele present for its corresponding phenotype to be observed. A dominant trait is represented by a capital letter in the genotype. With our example of height, tall is our dominant trait represented by a capital T. The letter used to represent a gene is typically the first letter of the dominant trait. The next term is a recessive trait. A recessive trait is one that needs both alleles present in the gene to be recessive for its corresponding phenotype to be observed. A recessive trait is represented by a lowercase letter in the genotype. With our example of height, short is our recessive trait, represented by a lowercase t. The last two terms are homozygous and heterozygous. These terms refer to the genotype and whether both letters are capital or lowercase, or if it is the combination of the two. If homozygous, a genotype can be homozygous dominant, which means both letters are capital, so big T in our example. Or it can be homozygous recessive, which means both letters are lowercase, so little t in our example. If the genotype is heterozygous, then there will be one big T and one little t. The phenotype of the heterozygous individual will be the same as the dominant trait phenotype, because the dominant trait only needs one dominant allele to be physically represented, as I explained earlier when we were talking about phenotypes. Now we can learn how to use a Punnett square. To begin, we must first draw out our Punnett square. It should look like a four-square chord, or a big square divided into four little squares, like shown here. Now we need the genotypes for the parents that are breeding. For this example, we will say the father is homozygous dominant, or big T, and the mother is homozygous recessive, or little t. Now place the father's genotype at the top of the square like shown, and the mother's genotype on the left-hand side like this. To complete the Punnett square, you need to fill in each square with the right cross of genotypes. The top right square will be our first square. The father gave a big T and the mother gave a little t. Write both of those letters in the box to determine the first possible outcome for an offspring for these parents. According to this, the child will have a heterozygous genotype that will show the dominant phenotype of tall. Follow the same process to complete the last three squares. You should end up with all four boxes filled with a big T and a little t, or a heterozygous genotype. Now let's do another example with a different set of genotypes. This time, both parents are heterozygous for the tall gene, meaning that the father and mother will both have a genotype with a big T and a little t. So we are going to set up our Punnett square just like we did last time with 4 squares. At the top, we will put the father's genotype, a big T and then a little t. And on the left-hand side, we will put the mother's genotype, a big T and a little t. Starting with the top left box again, let's fill them in. We first get a big T from the father and a big T from the mother. This genotype gives us a homozygous dominant, meaning the phenotype will be tall. Let's move on to the next box. The next box to the right is a little t from the father and a big T from the mother. This individual will be a heterozygous individual just like the parents, meaning it will also have a dominant phenotype of tall. And the third box in the bottom left will have a big T from the father and a little t from the mother. This will be the same as the last one, a heterozygous individual with a dominant tall phenotype. For the last box, we get a little t from the father and a little t from the mother. This individual will be homozygous recessive. This will be the first individual that we see that shows the short recessive phenotype. For the last box, we get a little t from the father and a little t from the mother. This individual will be
Powered by video.